All the cells that come out of meiosis 1 will now enter the second division, so meiosis has two divisions, called meiosis 2. So these were the cells at the end of meiosis 1, and they will both then undergo meiosis 2, and you will end up with 4 cells at the end of it. So there will be a prophase 2, metaphase 2, anaphase 2, and telophase 2 to end up dividing both of these cells once again. Now the point of this meiosis 2 process is separating these sister chromatids from each other. So you have double-stranded chromosomes that end up becoming single-stranded, because at the end you can see single-stranded chromosomes. So meiosis 2, the cells are all still haploid, chromosome number does not change, and it involves the separation of sister chromatids. So instead of halving the chromosome number by separating homologous pairs, like in meiosis 1, you separate the sister chromatids so that you go back to having single-stranded chromosomes, like is the natural state for your cells. And remember, this occurs in both cells produced in meiosis 1. So many diagrams will only show one cell undergoing prophase 2 or metaphase 2, but it is important to remember that just even if there's one cell, it could still be meiosis 2 that is taking place. So let's start with prophase 2. So prophase 2 is very similar to prophase 1, except because there are no homologous pairs, there therefore is no crossing over or the lining up of homologous pairs. So instead, the nuclear membrane and nucleolus will disintegrate, which you can see by the dotted line, and there's my nucleolus. And then the centrosome will still split into two centrioles, and the centrioles will move to opposite poles. So I showed this slightly differently to prophase 1 because I showed the centrosome splitting, but I have showed you here that the spindle threads or spindle fibers start to form. It's important to note that in this diagram, in this phase, each chromosome is visible as two chromatids joined by a centromere. So that would be one chromatid, that would be the other, and the little black dot is the centromere. So those chromosomes are all double-stranded. Let's move on to metaphase. So in metaphase 2, it's a similar arrangement of chromosomes. Metaphase, think middle of the cell, where the chromosomes line up at the middle of the cell. But instead of having pairs of chromosomes like in metaphase 1, which you can see very clearly in this diagram, you can see the homologous pairs lining up at the equator. Here, the chromosomes arrange themselves in a single file line, or we say they arrange themselves singly at the equator of the cell. So they're no longer these pairs on top of one another, it's singly spaced chromosomes because the pairs have been separated. And then the same thing as metaphase 1, chromosomes attached to the spindle by their centromeres. Once metaphase has been completed, now we separate the sister chromatids from each other. So in each of the cells, the spindle fibers will contract, which will separate sister chromatids because that chromosome was double-stranded. Now one chromatid from each of the chromosomes will go to each side. Then the chromosomes or chromatids are pulled to opposite poles. In this case, because they are separated, you could call them chromosomes, but in anaphase, it's still all right to call them chromatids. So just remember that anaphase 1, on the other hand, had the separation of homologous pairs. So those homologous pairs split and whole chromosomes were pulled to the poles. In that case, you can see that there are double-stranded chromosomes being pulled to the poles, whereas here, the chromatids have been separated, so single-stranded chromosomes are pulled to the poles. Now we enter the last phase of meiosis, which is telophase II. Here you can see two groups of chromosomes that would form at each of the poles in the cell. So that was the one cell that was dividing. This is the other cell. So two groups appear, one at each pole, and the nuclei will start to reform. The cytoplasm will divide in both the cells, and that's cytokinesis, and you end up with 4 genetically different cells being produced. All the cells that are produced are haploid, just like they were at the end of telophase 1, but instead of having double-stranded chromosomes, if you think back to what telophase 1 looked like, with double-stranded chromosomes in each nucleus, now there are single-stranded chromosomes in each of the cells. So there still are 3 chromosomes like there were at the end of telophase 1, but instead they are now single-stranded chromosomes. So as you can see, every single one of these cells that are produced through meiosis, remember these are gametes, so these could be sperm cells or egg cells, but if these were human cells, there would be 23 single-stranded chromosomes in each of them. Here there are only 3. So if you're asked, is this a human cell? No. Nobody's got time to draw 23 chromosomes. But I could ask you, how many chromosomes would be present in each nucleus if it were a sperm or an egg cell of a human? So then your answer would then be 23. So all these cells are completely different. These will have 2 paternal chromosomes, 1 maternal, but even those paternal chromosomes are different. And it is important that you understand the processes that give rise to this variation in the cells, crossing over and random arrangement of chromosomes. Crossing over happens in prophase 1, random arrangement of chromosomes in metaphase 1 and 2. And it's thanks to the differences in these gametes that gives rise to the variation that we see in living species.